Greetings again, Pencrest High School students. I met my limit for 15 minutes of talking about DNA, so I guess things got a little bit more complicated than I was thinking. So I had to make part two of the video. But there is one good thing. I noticed during translation that my face was actually covering the diagram for translation. So lucky for you, I got my face out of there so you can actually see translation going on. So if you take a look again at this picture. Down at the bottom there, you can see a couple things happening. Okay, the one thing you can see is you have that pink acorn there. That's the ribosome. The red ribbon at the bottom, that's messenger RNA. That's what we made in the nucleus by transcription. Okay, each three bases on the messenger RNA are called a codon. Okay, that cross that's coming down, it almost looks like a hairpin. That's transfer RNA, and it brings bases that bond to the messenger RNA. I know it's a lot of vocabulary. The three bases on transfer RNA are called anticodons. But look, transfer RNA just doesn't carry anticodons. It is also carrying that little bluish green sphere on the top of it. That's an amino acid. Okay, so anywhere between like thousands and tens of thousands of these amino acids connected together will actually make a protein. So if you're confused, lost, or scared with all of the written stuff on that slide, let's try to make it a little bit easier and we'll simplify it. Okay, this is a very simplified diagram. It looks like Tetris. It's a very simplified diagram of translation, but I like it because it's simple. Look at the bottom. That red ribbon is messenger RNA. Okay, that messenger RNA attaches to the ribosome. The ribosome is the gray peanut-looking thing. All right. Whenever we translate going from messenger RNA to a protein, the first three bases in every protein on Earth, it's AUG. It's the same codon. It's AUG for every single protein on Earth. But you can see those three bases on messenger RNA are going to match up with those blue bases on the blue rectangle. In this case, the blue rectangle is transfer RNA. It's just a different look at it. Okay? And you can see it's bringing the complementary bases into the messenger RNA. But on the back end, and in this picture, the circles are green. On the back end, it's carrying amino acids. Okay, and it's snapping them together. You might remember from our biochemistry unit that there's a special type of bond that connects amino acids together. That's called a peptide bond. Okay, so you can see these amino acids actually being strung together, and those are the molecules that actually make the proteins. And the proteins, as I said before, they're kind of like the bricks to the house. You're the house, and the bricks are the proteins. So it's like actually making the structure. So let's step back globally and just make sure that we know the difference between these two things. Okay, transcription is taking DNA and turning it into RNA. Okay? Translation, which occurs at the ribosome, is taking that messenger RNA and converting that into amino acids. Transcription takes place in the nucleus. Translation takes place at the ribosome. But wait, there's more. Scientists have actually figured out the sequence of codons on messenger RNA. There are 20 different amino acids, and they figured out which codons code for which amino acids. So if you take a look at this, let's think of our first three bases to every single amino acid. Or to every protein. It's going to be AUG. Okay. All you need to do, and I'm not going to make you memorize this or anything like that. You just need to be able to use the chart. So watch. First letter is A. Second letter is U, so where they meet is going to be this box here, and then the last letter is G. A, U, G. The amino acid for all proteins, the first one is always going to be methionine. Scientists usually just use the first three letters of an amino acid because you don't want to write things like lysine and isoleucine for thousands and tens of thousands of amino acids. When the protein is done being made, take a look at this. If it runs into the codon UAA, it doesn't code for an amino acid. It says stop, which means stop making the protein. The protein is made. We can send it out to either the Golgi apparatus or we could send it out of the cell. If it's something like keratin, maybe it could go to your fingernails. If it's hemoglobin, it'll stay in the blood. Okay? So here they are. No need to memorize them. I just want you guys to see what they are. Okay? There's the 20 different amino acids. The thing that's amazing is that it's these 20 amino acids that make up the proteins for every living thing on Earth. Banana slug, rhinoceros, maple tree, Mr. Dell. It's the same 20 amino acids. They're just arranged differently. So imagine if you just had 20 different colors of Legos, and you had all had to make something. Everybody would make something different, even though it contains the same 20 colors of Legos. It's the same thing with amino acids, which is really quite amazing. And for those of you guys who want the bigger picture, remember it's suggesting too that all life here on Earth is actually related, because it's a blade of grass that still has those amino acids in it, the same as a giraffe, which is pretty amazing. So look, this is going the whole way then. Ready? If you start with DNA, you can take DNA, turn it into RNA, and turn the RNA into a protein. I'm not going to bore you by going through the whole line here, although I will say this. If we take this first DNA, the first 3 bases, and I convert it to RNA, so I'm just going to do this right here. Ready? A bonds to, this would be a U, A bonds to U, T bonds to A. So I'd have U, U, A. Look over here, U, U, A. There it is. I see LEU. I would go back and I'd be like, oh, LEU, it's leucine. I'm not going to make you write out the whole amino acid, just the first 3 letters, because that's how scientists usually do it. So look, I went from DNA, I transcribed it to messenger RNA, and then I translated it into amino acid. Now, you need to get a whole bunch of those amino acids connected together to actually make up a protein. This is just another close-up picture showing you the difference between codons and anticodons over there on the left side. You can see the ribosome, but this gives you a really nice look at the 3 bases on messenger RNA, which are the codons. That would be yellow, and then, I don't know, it looks light green to me. Transfer RNA is going to bring in those anticodons, and then on the back end, it'll bring the amino acid. You might remember from earlier in the year, we had this discussion about what makes you look young and me look old. And then one of the big ones is collagen, right? Collagen is the most abundant protein in the body. Look, you can actually see the amino acid sequence for collagen, okay? Glycine, proline, alanine, and you can see that it actually makes like a legitimate structure. This is a computer simulated structure of collagen. It's a triple helix. It's 3 ropes that are like twisted on one another. There's all different types of collagen out there. There's different types of collagen for different animals, but there it is. And look, the amino acid sequence for collagen, it's a little over 1,000 amino acids connected together. Just to put the nice little bow on this, remember that all things on Earth are related to one another. That process of transcription and translation occurs in every living thing on Earth. So an elephant, a maple tree, a bacteria, yes, and I found an old picture of Mr. Virtue when he was in college, so I decided to put that in there for you too. Yes, transcription and translation even occurs in Mr. Virtue. I hope that me going through this gave you a little review, or maybe I was able to clarify some things when it comes to DNA. And I'll see you on the other side. Thanks for listening.